MIT's new approach to gene editing is really a game changer. They've refined prime editing to be 60 times more precise, reducing errors drastically.
That sounds impressive. A person,
Exactly. They've modified the Cas9 protein, the one that guides the editing process, so it cuts DNA more precisely, almost like recalibrating a fine engine.
yeah. So this isn't about creating a new tool from scratch. It's more about like remixing what we already have with prime editing.
Right. They took the existing prime editing system, which already avoids those risky double stranded cuts, and then further tweaked the enzyme. This new technique reduces unintended mutations, bringing down the error from, say, 1 in about 7 edits to 1 in over 100.
That's, wow, turning a roughly 14% error rate into less than 1% in some modes. Person to,
Yep, and it's like, imagine you have a machine that's making mistakes every seventh time. Now, with this upgrade, it's almost as if that same machine only messes up every 100 or so times. It uses mutated versions of Cas9 to destabilize the old DNA strands and let in the corrected new ones.
I do get the concept, but don't you think tweaking the enzyme like that might affect the overall efficiency of the edit? I mean, sometimes precision comes at the cost of speed or yield.
That's a fair point, but MIT's method keeps the delivery system simple. They aren't adding extra steps, just fine tuning the process. So, while there might be concerns initially, early tests in mouse and human cells showed it's both safer and still efficient.
Right. Interesting. And they also mentioned, if I recall correctly, that they improved the RNA binding component to better stabilize the RNA template, correct?
Yes, exactly. They incorporated an RNA binding protein to secure the template, which is critical for ensuring the new sequence gets inserted correctly. This little tweak helps in making sure that the unwanted mutations are nearly eliminated.
And this is all done without like making a full double strand break in the DNA. That's a huge safety advantage.
Precisely. It's all about minimizing those dangerous double cuts. Instead, the system performs a gentler, single strand cut, opening a tiny flap where the correction is inserted. It's a bit like carefully replacing a page in a book without ripping out the whole chapter.
That's a neat analogy. So, with these innovations, could we see a leap in treating genetic diseases?
Absolutely. Diseases that were once too risky to target with gene therapy might finally be within reach. This tool could, in principle, repair faulty genes across a wide array of genetic disorders.
While it's very promising, I do wonder about long-term studies. It's one thing to see success in cells, it's another to translate that into clinical practice without unforeseen issues.
True, long term safety will always be the watchword. But as Robert Langer, one of the senior researchers, pointed out, the goal is to have an effective treatment with minimal side effects. And this refinement makes that a lot more achievable.
Yeah, it's fascinating how this builds upon decades of gene editing, from viral vectors to zinc finger nucleases to CRISPR, and now this ultra-precise prime editing upgrade.
Definitely. The evolution is remarkable, and MIT's breakthrough not only enhances safety, but also paves the way for integrating these editors into broader research and, eventually, therapeutic contexts.
It really emphasizes how science is continuously pushing boundaries. I'm excited to see how these advancements might one day transform medicine completely.
Me too. In a nutshell, this innovative approach could be the key to unlocking safer, more reliable gene therapies that address hundreds of genetic disorders without the typical risks.
And while challenges like efficient delivery to specific tissues remain, the progress is nothing short of revolutionary. Thanks for this deep dive.
Absolutely. It's like upgrading the engine of gene therapy to a precision tuned marvel. Here's hoping the next steps bring it even closer to clinical reality.
For sure, this breakthrough shows how a few strategic tweaks can make all the difference in the world of medicine.